Today we will continue our discussion about how this KRAS mutation can lead to the PDAC migration. So we will now discuss some of the experimental data that have been published in the paper. So this is the experimental overview that is presented in the panel A, where they took the mice and when they reach up to this 18 week of the and that does not have any pancreatic disease, they have the healthy pancreas, and thereafter they induce this KRAS mutations with the help of doxycycline, as the KRAS mutations are doxycycline driven that are been injected in these mice. So after that they waited for the 6 weeks and then stopped injecting these KRAS mutations, and then they collected the data at 5 day, 2 week and 4 weeks. And simultaneously, what they have observed, when the doxycycline has been induction has been stopped, they can see that tumor regressions has been shown within the pancreatic within the mice. So this is the experimentals that they did. Now let us understand what are the data that they have occupied at this 5 day, 2 week and 4 week of the doxycycline injections has been stopped. So when they check the immune cells data, that is cells that is related to the CD/T plus cells, and importantly they identify the CD45 plus different subpopulations of the CD45 plus cells. At the same time, they also check these myeloid cells, which helps in the tumor progressions. And when they compared between this KRAS on and this KRAS off at 5 day, they can see that the number of immune cells that are present in this KRAS in which the doxycycline has been induction has been stopped are more. That is CD45 plus CD45 plus cells subpopulations are more in the case of this mice that has this KRAS on, and in the case of mice in which this KRAS is been off, that can be seen here. That is CD3 plus T cells and other CD45 plus populations. And when they check this, when they checked about these myeloid cells, that is CD11B positive and CD11B positive lyse6 positive lyse-G positive other myeloid subpopulation cells, and they can see that these tumor progressions immune cells are more in the case of mice where this KRAS is on, compared to the conditions where KRAS has been off by 5 days. And when they quantify these things in this panel D, they can see that the tumor suppression immune cells are been increased in this KRAS 5 day off samples data, and whereas these myeloid populations are myeloid subpopulations are more in the case of this myeloid subpopulations are also reduced in the case of this when these tumors are being compared between KRAS 5D and when this KRAS is on the mice models. So these are the this panel D shows the fold change in the case of this KRAS 5D when it's compared with the KRAS on mice models, and we can see that there is an increase in the case of these immune suppression cells, and there is a decrease in the case of this immune progression or immune cells that are being differentiated themselves towards supporting immune progressions. And there are different subpopulations that are shown here. And when they check these immunostainings, and they can see that when this KRAS is off, so these are the controls first four figures, and when the KRAS is off, they can see that increase in the amount of CD4 cells, and the CD8 cells are in low amount. CD8s are, you can consider them as here as a tumor progression cells, and green cells are tumor suppression cells. So after 2 weeks, they can see the increase in the amount of this tumor suppression immune cells, and after 4 weeks they can see the same similar increase in the amount of CD4 plus, or the CD4 plus cells are sustained compared to this 2 weeks. However, when they check for CD11, that is myeloid cells that helps in the tumor progressions, uh, in the in the case where the KRAS is on, and when the KRAS is off, but after 5 days they can still see this myeloid cells are still there. However, after 2 weeks and 4 weeks, they can see decrease in the populations of these myeloid cells. The tumor progression cells are reduced. When they quantify these things, they can see the similar things. They can see the similar observations, that is CD4 plus cells are more in the case of when the KRAS is off, compared to the case where the myeloid populations are more when the KRAS is on. So this indicates that the KRAS related mutations affect the immune efficiency, and this is the further quantifications, that is when the KRAS is on, how this what are the kind of immune cells that are being identified. And we can see that the 67% of the cells are these myeloid cells, whereas when this KRAS is off by the 5 days, they can see that 38 it reduced to 38%. And they did the experiment on PIKT mouse models also, and there also they can see the decrease in the myeloid populations from 75% to 45% in just 5 days after the KRAS is off. And panel HIJK is just the quantifications of the compositions that are presented in this panel G with the help of flow cytometers. So in this case we can see that CD4 plus cells are high in the case of this when the KRAS is on, whereas the CD8 plus cells are high when the KRAS is off. Then these data are about these data are related to the 5 day off data. So here also the CD4 plus cells here is the subpopulation that represents the CD4 plus cells in this panel is related to these myeloid cells populations. And after this in the panel G, here we can see that in the y-axis it shows this TM3 plus percentage, and in the panel I we can see it's the TBET percentage, and here we can see that in the when the KRAS is off, these percentages higher, and these have tumor suppressions effects, whereas TM3 plus has the tumor progressions effect. Then thereafter they check this relative expression of this relative expressions of the immune cells, that relative IFNG expressions through flow cytometers, and they can see that when the KRAS is on, it's almost negligible compared to the case when the KRAS is off. And similarly, when they check the CD 86 plus percentage, and they can see that they are high in the case of this KRAS 5D KRAS mutations are off, and similar things has been observed for the CD44 plus populations also. So this indicates that all the tumor suppressions, this data indicates, or this data and figure indicates that all the tumor suppressions immune cells are high when the KRAS is off, or when the KRAS is eliminated. Whereas when the KRAS is on, they can see that tumor progressions, immune cells, or immune cells that are differentiate themselves to help in tumor progressions are high. So this indicates this figure made a hypothesis that it's the KRAS mutations that affect the immune efficiency, and if KRAS mutations are being somehow eliminated from the pancreatic cancer cells, then it will increase the immune function in the host to act against the pancreatic tumor cells. We will continue these discussions and discuss the other figures of this paper in the next video. So that's all for this video, and if you need to understand more about this experimentals procedure, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.